Now the question is, how can you actually understand such a process? And this is essentially what you need to do, is you need to use the individual components and you have to reconstitute it, you have to reconstruct it in the test tube. You have to basically take the parts and then, show that, let's say, part 1, 2, 3, 4, 5, or you are suspicious, you put them together and analyze whether they can actually do the job you're interested in. This is not easy, but this is exactly a challenge which this year's prize winner, Thomas Wollert, has taken on. And he has not only taken it on once, but twice, and I will tell you about it. Now, a little bit of background on Thomas. And so I'm disclosing now some of your information you gave me on the telephone. I hope you don't mind. So Thomas is a biochemist by training who did his undergraduate studies in Potsdam and then moved to Hanover. And he told me on the phone that Potsdam was nice, but there was so much plant biochemistry, and he was not that enthused by plant biochemistry at that time, which I kind of sympathize with, but is there a plant scientist here? Hopefully not. Anyway, so he moved to Hanover, and one of the reasons also is that it was a more physical or physical-chemical curriculum, which attracted him. And he was soon being pulled into structural biology, crystallography. Then he moved to the Helmholtz Center of Infection Biology in Brunswick, where he then, in his PhD thesis, where he worked on the structure of Listeria proteins. And Listeria is a pathogenic bacterium, which you find it sometimes in cheese, and it can enter cells, and it zips around in cells and can do damage. The problem at that time was that it's usually when you study these organisms, you want to have an animal model, so you want to use mice, but these guys didn't infect mice. And the reason was simply that the receptor on the cell surface, which binds to a Listeria surface protein in the human one, the catecholamine receptor, has a high affinity for the bacterial protein, but the mouse receptor doesn't. So what Thomas managed to do is crystallize, got a crystal structure, and from the structure, he was actually able to design a slight modification of the protein, which increased the affinity by, I think it was 10,000-fold. It was amazing, 4 orders of magnitude, which is very rare, I should say. And with this increased affinity, the affinity was high enough for infecting mice cells, and so far now mouse model systems can be utilized. And that was quite a breakthrough, his first discovery, I should say. Now, at that time, when you complete a PhD, you want to look around, and Thomas is a very entrepreneurial person. He didn't want to stick in the field, and he also told me that he was a little bit influenced, I should say, by myself. I didn't know that. He listened to a seminar of mine and learned about membranes. I'm a membrane person, so he got fascinated with membranes. And then he also looked around and listened to a talk of Jim Hurley, at that time at the NIH, who works with protein-membrane interaction, actually membrane remodeling by proteins. And that fascinated him, also being firmly rooted in structural biology, and he applied to his lab and eventually joined his lab. Now, I told you already that Thomas has very much his own mind, which is if the young scientists are here, having your own crazy ideas and sticking to them is not necessarily a bad idea, but I'll tell you. So he was assigned a project. The project was something which didn't quite suffice him. It was not challenging enough. So it actually dealt with this second garbage collection processing pathway, where you form these vesicles inside vesicles. And Thomas said, well, if I work on this, why don't we just reconstitute the goddamn thing, which nobody had done before. So just, take purified components, which were pretty much available, they were crystallized. So the protein chemistry was well worked out, but nobody had ever really put everything together, put in a big artificial vesicle and put these proteins on and see whether they can force these proteins to actually generate vesicles, which are then kind of pushed inside these giant vesicles. I'm not sure what Jim Hurley, he was apparently not so enthused, and he probably thought this young German postdoc must still arrive in reality, This is insane, But he didn't stop him. So Thomas was allowed to, roam around at the NIH, work in another lab of Jennifer Lippert-Schwartz to learn giant liposomes. And to cut a long story short, he actually pulled it off. And that was quite a splash. It was a big paper. I remember that. We talked even at that time. It was amazing discovery, and I think Jim's opinion, as always, when we think this is not going to work, and when if then someone in our lab pulls it off, we're obviously very excited. And so that was the second success story. But that's not enough, because Thomas then was selected as a Max-Planck Junior Group leader, moved to Munich, and he again did something which 9 out of 10, I would say, even 95 out of 100 would not do. When you do a successful postdoc in a different field from your PhD, and you get your own lab, what you do is you continue on that line. you do the same thing. You just expand on that. That's actually quite natural, quite normal. He said, no way. I want to do something else. So he didn't want to jump ship totally. He said, well, I maybe want to start working on reconstitution again. But he then thought about the third garbage collection pathway, the autophagocytosis, which is even much more complicated in a way. He thought, why don't we try to reconstitute that one? And he put really all the eggs in his basket, just, not fuzzing around with, little things, not publishing much for a while, to try to get everything together. And I wouldn't be standing here and telling you about Thomas's work if he hadn't been successful as well. So I have to say, this is really quite amazing. The jury was very deeply impressed by looking at his record. Someone who 3 times in his career has done really quite remarkable work, at a relatively young age. This is something you can be congratulated for. I think it also deserves a bit of admiration that someone is really capable of doing such a thing. And it's really with my utmost pleasure that we have been able to select Thomas Wollert as this year's winners of the Eppendorf Young Investigator Award. Now, Thomas, I would like now to step up here and accept the award. So first of all, you get this very beautiful, first of all, let me just stand you here and congratulate you. And you get this. Yeah, we're not done yet, because there's a second part. I just take it for you. Up here, you will speak anyway. So there is this, the documentation here, I don't know how you say Urkunde in English. Certificate. And that's yours as well.
Can you both have it?
Yeah. Okay, so welcome Thomas Wollert, and we are now very much looking forward to your lecture.
So, thanks for this very kind introduction. So that was a very special introduction. I never received that before. So thank you very much. And so I will, first of all, I would like to thank Reinhard for introducing my work, and also the jury for selecting my work, and also, of course, Eppendorf and Nature to collaborate to make this here to set the stage, especially for me, to present my work. And I'm very happy to do that tonight. And I'm also very happy to be here at the EMBL, of course, which is a great place for science, and to get the chance to talk about the own science here in this audience is a real pleasure. So let me just start with me hiking in the mountains. So if you want to do that as a human, what you need is a couple of specialized cells, which are, for example, neurons, which are coordinating the movement of our bodies, and some muscles, which are making us move, and epithelial cells that are covering and sheltering our bodies from the environment. And they are quite different in structure and shape, but if you have a closer look, they are really conserved. If you look at the cellular organization, this cell is composed of the cytoplasm, which is like an aqueous environment, and it's organized into, or compartmentalized into, organelles, which are membrane-bound compartments. And they have very special functions. For example, mitochondria are the major power plants of our cells. They are generating energy. And the endoplasmic reticulum here is a major biosynthetic organelle. And they are highly specialized, but they exchange material, for example, here with the cytoplasm, and both of these organelles are exchanging quite vividly with the cytoplasm. But if they want to be transported further, reach other organelles, they take a specific pathway. And it was a challenge in biology some decades ago to identify this pathway. And it has led in 2013 to the Nobel Prize to discover this pathway. These three gentlemen have identified the major principles which are involved in bringing proteins from the cytoplasm actually to the plasma membrane. But as Reinhard has already alluded to, there's another organelle, And this organelle actually receives material from the cell and degrades this material. And this is important to maintain homeostasis. You don't want to accumulate garbage in the cell. So there is another access route, and almost every part of the cell can have a direct linkage to lysosomes and to deliver material to be degraded there. And when I started my lab, I got interested in this access route because it involves a very special pathway, needs to be highly selective, but it needs to deal with a broad range of material, which are small components, big components, and so on. So, and I'm more than happy that I was awarded this prize because it gives me the audience to present our work here, but it's not my effort over the past 5 years. It was really a team effort of my lab, and I'm more than happy that I have this team working with me together to reconstitute complex molecular machines in the test tube, which is challenging. And they are very talented and also living for science like I do. And the major pathway we are investigated is a recycling pathway. And this recycling pathway basically is not only responsible for maintaining cellular homeostasis by degrading material which is damaging the cell, for example. It's also induced if the cell encounters stress conditions, such as starvation, for example. So then this pathway can utilize or recycles just bulk material and then makes building blocks available. And those building blocks are then used by the cell to maintain vital functions. It's also involved in differentiation and in immunity, and for that reason, there are a couple of human diseases connected to this pathway, and that makes obviously the pathway quite interesting. It's the stress response pathway is connected with cancer, for example, homeostasis with neurodegenerative disease, differentiation with anemia, and immunity, obviously, with some sort of autoimmune diseases. So to understand the pathway would allow us to also interfere with those diseases. And we chose the yeast as a model organism. That is the most simplest eukaryotic cell you could imagine. But coming back to my introduction, the general architecture of the yeast cell is the same as in human cell. So you can identify the general pathways in yeast to get into the major function of those pathways, and then you can look what is different in humans and why have humans acquired a variety of variations of this pathway. So it's a quite useful model organism, and in yeast, how this works is that a couple of small vesicles, probably derived from the Golgi, but it's still a matter of debate where it's coming from. They are coming together and they are fused to give rise to this cup-shaped membrane here. And this can then select cargo, and this selection could happen either selectively, that means it's only a material targeted to degradation, which is enclosed by this membrane, which maintains cellular homeostasis, and it can also switch to non-selective capturing of material, which is then important to respond to stress conditions. So this switch is important to understand, and after that, the material is enclosed by the autophagosome, so there's an expansion step, the structure is then sealed, and then the lysosome is fusing with it, and the material is degraded. There are, at the moment, not 38, but 40 autophagy-related proteins which are driving this complex, so still in the process of discovering all the components, but the major components have been identified, and they are conserved again from yeast to humans, and somehow they drive initiation, expansion, and all the other steps. But how they are doing that was more or less unknown, and so what my approach was over the time, I and now we as a team try to purify all the components, assembling the functional complexes, the macromolecular machines from them, then reconstituting them on artificial membranes, on model membrane systems to see what their action is in vitro in the test tube, and then we go back in the in vivo system to study their action there. And sorry, this is basically to highlight the important steps. And first of all, if you are concerned in manipulating the pathway, you could think about increasing the number of autophagosomes. But for that step, you need to understand how initiation takes place. And that was one project we did in the lab. It was known that there is a big complex called ATG1 kinase complex, which assembles up an induction of this pathway. And it somehow crosstalks with the starter vesicles, ATG9 vesicles. And they are labeled with a transmembrane protein called ATG9, which marks them as being a source for autophagosome formation. And the question was how they are doing that, and we started with reconstituting those complexes. ATG1 kinase complex is roughly 600 kilodalton, and this is here 6-transmembrane helical protein. So we got all the components, we put them together, and then we studied their interaction. And here's just the architecture of this huge complex which drives this initiation. It's composed of 5 subunits, and those 5 subunits are usually present in the cytoplasm as inactive subcomplexes. And it's here this long scaffolding complex and a regulatory complex. And upon induction of autophagy, they are coming together, and then they are in their active form and initiating autophagy. So we went out to purify all the single components, and that worked quite well. So we got all the single components, put them together, get the 2 subcomplexes, which are inactive, and then we assembled the entire full-length complex. This is all purified components here. And we analyzed this again, and here you can see gel filtration profiles of this of the scaffolding complex and of this activator complex. And if we put both together, we can generate the full-length complex, and we characterized its molecular weight as roughly what we have calculated. So that worked quite nicely. And then we looked, okay, what happens now if we bring this complex together with its interaction partner ATG9 in membranes? And we looked for all the subunits and identified that this scaffolding subunit here is tightly interacting with ATG9, which is shown here in this experiment, but it's only recognizing the protein itself and not the membrane. And this is something special because usually these proteins have a structure which reminds on membrane interacting proteins, but this protein does not touch membranes at all. It specifically recognizes this interaction partner. And this is very important to have the control of initiating autophagy. And we then looked at our ATG9 proteoliposomes. They are 30 to 60 nanometers in diameter. This is here cryo-electron microscopy. And if we now mix them with our scaffolding component, we found that a couple of vesicles are stick together. So somehow this is a tethering complex, and it brings together 2 vesicles, which makes sense because it has 2 binding sites for ATG9. And then we went further and we investigated this inactive subunit, which has these 2 regulatory guys occupying this crescent-shaped structure here. And if we combine those with ATG9 proteoliposomes, nothing happens. So that means this is really switched off. But if we put together the entire complex, we regain all the activity, and tethering already takes place. So that means that we have an activation of this scaffolding component, which is inhibited by these 2 regulatory proteins, by this regulatory complex here. And this initiates or set up the machine to tether vesicles, and presumably this leads then to an interaction, or we've shown that this leads to an interaction with ATG9 to tethering. And in the end, it presumably leads to nucleation of this autophagic membrane, which probably involves proteins which are also investigated by Reinhard, that are the major fusion machinery, the snare proteins which are driving this process. Now, if we have understood this initiation process, there's also expansion. And you can also manipulate autophagy if you want to increase the size of this container or decrease it. So we wanted to understand this as well. And so in terms of regulating the size, to understand expansion, we need to know what's going on here. And there's this very special small protein. It's the smallest protein that is in the entire pathway. It's called ATG8, but this is a quite interesting guy. So this protein is covalently attached to the membrane of autophagosomes, and it's the most abundant protein there. And it drives somehow the expansion of autophagosomes, but it was not known how it's doing this job. It's just a small protein attached to lipids in the membrane, but what was all about that? It involves a quite complicated machine. It's similar to the ubiquitin conjugation machine, which is labeling proteins to be delivered to the proteasome, and Reinhard has already talked about that. And so there is an E1-like enzyme and an E2-like enzyme activating and transferring the small molecules to the membrane. And then there is a help of an E3-like ligase complex, a big complex, which is facilitating the final step of getting ATG8 to the membrane. And we actually reconstituted also this entire system. So here we used model membranes, which we visualized by putting a fluorescent dye in this membrane. So here it's giant unilamellar vesicles, which are visualized by confocal fluorescent microscopy. And we reconstituted this entire pathway, and here you can see fluorescent labeled ATG8 in a live imaged reaction, how it's getting to the membrane by this E3 ligase complex, which is also fluorescently labeled. And to our surprise, this complex stayed on the membrane after it has done its job. So why should an enzyme stay connected to its product? That was a little bit puzzling. But then we looked a little bit closer, and we realized that ATG8 is actually able to self-organize on the membrane, and it's even more interesting because it can stabilize various different membrane shapes. And that already suggested to us that it's maybe forming a container or some rigid structure which is guiding membrane structure. And so we looked further, and we used atomic force microscopy, which is a very high resolution technique to visualize molecules or membranes. And here we see real molecules, or the profile of real molecules on the membrane. And we discovered that there is a more or less regular arrangement of those structures on the membrane. It's just a single sheet of proteins which are assembled on the membrane, which are connected such that a membrane scaffold more or less is built. And that reminds us to like well-known systems like the Clathrin code, which is well studied and which is driving the uptake of plasma membrane and to bend the membrane here. So it's diverse pathways, but probably similar mechanisms. And so we think here it's ATG8 is forming the scaffolding and thereby driving the expansion of autophagosomes. So if we now compare that and bring this together with the general concept of vesicular transport, and we look what happens if you have this canonical transport processes. Their membrane is acquired from a donor organelle, and there is this well-studied system like Clathrin, COP1, COP2, which are coating protein codes which drive this reaction and bud of vesicles. And then they are brought to their destination organelle, and then there are tethering compartments or components which mediate the first contact to those vesicles, bringing them into close proximity with the membrane to allow their fusion, which is driven by a dedicated fusion machinery. And if we now compare this to autophagy, we could show that in this initiation step, there is some similarity because vesicles need to be brought together, and this is mediated by a tethering complex which we identified. And there is an expansion step, which is then driven by a membrane scaffold, which has similarities to canonical coating structures in cells. So we end up with a surprise because we have very diverse transport mechanisms in cells. Obviously, morphologically, they are quite different, but they share the same molecular machinery. And that was really interesting, and we are happy to follow up on that. And what has it now to do with biomedical impact and with biomedicine? If you think about neurodegeneration, the problem for neurons is that there are a couple of bad proteins which tend to aggregate, and they form this insoluble bigger arrangements which are toxic for the cell. In cancer, you can think about of treatment. In chemotherapy, you want to damage the cell as much as you can, to accumulate as much as cell damage as you can, to force the cell to die. And basically, autophagy is acting in both pathways. For the protein aggregates, it's beneficial because it can remove those materials. So it's not preventing neurodegeneration by preventing the accumulate or the aggregation of proteins, but it is able to remove and to deal with those components in the end. In cancer treatment, it is important for the cancer cell to survive because it can get rid of damaged material. So here it's not beneficial for us as humans to have the system running. So if we know to manipulate the pathway, we get maybe a handle on treatment options, which is here increasing the throughput of cargo in neurodegenerative disease to even be able to revert accumulation that has occurred in the past. And in cancer, presumably or hopefully, one can downregulate autophagy to make this chemotherapy much more efficient. And there are some indications that this is really the case. And with this, I would like to thank my research team, of course, which allowed me to get started with this endeavor to finally reconstitute the entire pathway in the test tube. Hopefully we are there in 10 or 20 years. And our collaborators, I'm very happy that I could share equipment, for example, from Petra Schwilles' lab, the atomic force microscope. We exchanged material with Roland Wedlich-Söldner's group, Julia von Blume, Stefan Jentsch. And I have also collaboration for structural biology with Christine Ziegler. And of course, all the funding bodies, without money, you can't do anything, and research is quite expensive. So we are happy to have acquired a couple of funding sources to allow our lab to do this research. And thank you for your attention.